Gregor Mendel was an Austrian monk. He studied at the University of Vienna from 1851 to 1853, where he was influenced by a physicist who encouraged experimentation and the application of mathematics to science, and a botanist who aroused Mendel's ideas in the causes, interest at least, in the causes of variation in plants. After university, Mendel taught at the Brunn Modern School and lived in the local monastery. The monks of this monastery had a very long tradition of interest in the breeding of plants, including peas, and around 1857, Mendel began breeding garden peas to study inheritance. Now, people had studied peas before, and they had tried to study inheritance before, but Mendel used the scientific method. He used a very good experimental method. He used math. He used statistics to come to an understanding that no one else had found. Mendel picked the pea plant because he could control the breeding. If you actually look at what he did, he removed the anthers, which he removed the sexual organs basically from the flowers so that he could physically control the pollen transfer. So when he was doing these studies, he had the parent generations, which he labeled P, and then the first children generation, the first filial generation, F1. So when you make an F1 generation, you're breeding two parents. When you want to make an F2 generation, you breed two F1s. And so that's how he studied this. But by removing the anthers, he was directly able to control the breeding to know what bred with what. It wasn't left up to random chance. It turns out that pea plants are actually very good for genetic research. They're available in lots of different varieties with very distinct traits. They had strict control over which plants mated with which. Mendel could cross-pollinate them. Turns out they were relatively simple genetically. Most characteristics were controlled by a single gene, and each gene only had two alleles or two forms, one of which was completely dominant over the other. And that's not typical of most organisms. They're not normally that cut and dry, but turns out that Mendel was actually pretty lucky with his choice. So Mendel actually collected data on 7 different characteristics in pea plants, each with 2 options, each with 1 dominant option and 1 recessive option. When he actually looked at the ratio in the F2 generation, he found a 3-to-1 relationship overall. Now it is believed that these numbers are a little too perfect. He may have manipulated them in some way, but as his notebooks were burned when he died, we don't really have any way to check. Just as a quick reminder, please remember that genotype is the genetic makeup, and phenotype is the physical expression of a trait. So I had my purple flowers, and I had my homozygous dominant purple flowers. So homozygous dominant is genotype, and purple is phenotype. So you use a Punnett square to predict the ratio or percentage of the possible genes that an offspring will have based on the genotype of the parents. You put one parent on the top, one parent on the left, and you merge their results. In this case, I am taking a homozygous dominant parent and a homozygous recessive parent and breeding them together in my Punnett square to produce an F1 generation that was 100% heterozygous. If you then breed 2 F1s together to produce the F2, you can start to see what Mendel saw. He saw a 3-to-1 relationship with 3 dominants and 1 recessive. So in this case, I would have 3 purple offspring and 1 white offspring. Now he could only see phenotypes, but we also need to be aware that genotypes are involved here. Genotype are the genes that you actually have. That would be your homozygous dominant, heterozygous, and homozygous recessive. And phenotype would be the physical expression, in this case purple and white. So looking at this, I have a 1-to-2-to-1 relationship in my genotypic ratio, and I have a 3-to-1 relationship in my phenotypic ratio. If I breed 2 heterozygous parents in a monohybrid cross, or the traditional 4-square Punnett square, I will always see these ratios. When you actually look at the traits that you inherit, we see that there are alternative versions or alleles. We've seen that already in that there are purple and white flowers. Now, Inherited traits are just characteristics that are passed from parent to offspring, and they do that by passing on chromosomes. So when you actually look at the chromosomes that an organism gets, we see that you have 2 copies of every chromosome. Why does that make sense? Well, you got 1 copy from your dad, you got 1 copy from your mom, so you have 2 copies of each chromosome. We as humans have 23 pairs. Okay, so 46 chromosomes, but 23 from mom, 23 from dad. Now, some of those traits mask other traits, and we call those dominant alleles. They are always represented as capital letters to show the difference between dominant and recessive traits. And when you are determining the letter that you are choosing, it should be based off of the dominant trait. The recessive trait will be hidden by the presence of a dominant trait. So the only way you'll show a recessive characteristic, say having blue eyes, is if there are no dominant alleles in the way. Now, please be aware that just because an allele is dominant does not mean it is better. Does not even mean that it's more common. It just means that its expression is going to mask a recessive. Now, Mendel turns out actually got very lucky with his choices, as we've said previously. Traditionally, alleles that are near each other on a chromosome tend to be inherited together. We call that linked. Mendel studied 7 different traits, and statistically speaking, some of those traits probably should have been linked. But what he found indirectly was that these traits were either on separate chromosomes or far enough away from each other that they were not considered linked. Due to dominance, an organism's phenotype can be determined by multiple genotypes. A heterozygous purple flower is going to be just as purple as a homozygous dominant purple flower. So if you want to determine the alleles that something is carrying in their genotype, you need to breed it with a homozygous recessive. So in this case, if I know that I have a purple flower, I'm going to breed it with a white flower. If I get offspring that are all purple, I'll know that I had capital P, or homozygous dominant. But if I get some white offspring, I will know that my unknown was heterozygous. Mendel actually developed 2 laws based off of what he saw. The law of segregation says that when gametes are produced during meiosis, homologous chromosomes separate from each other. So it's called here the principle of segregation. Each allele for a trait is segregated into separate gametes. So when you have 1 copy from your mom and 1 copy from your dad, 1 copy is going to go one direction and the other copy is going to go another. So that means that you're going to separate those things. Now, the law of independent assortment talks about when things are lining up in metaphase. So if I have mom's version for chromosome 1, 2, and 3, and dad's chromosomes 1, 2, and 3, which order and which side mom's goes on and which side dad goes on is completely random. Now, this is only going to be true for genes on separate chromosomes. If they are linked or they are on the same chromosome, they will be inherited together. So far we've seen monohybrid crosses, or the traditional 4-square Punnett squares, but that's not really very useful in most situations. You need to be able to move on to further levels of complexity. The next step is a dihybrid cross, where you actually have crosses where it's 4 by 4. Now, to do that, you first need to find gametes. Now, there's 2 ways to do that. You can foil them, or you can use a monohybrid Punnett square, just sort of modified with 1 letter in each box to produce my 4 gametes. So in this case, I am looking at capital Y, lowercase y, capital R, lowercase r. Now, I know, based off the formula 2 to the n, where n is the number of heterozygotes, that I have 4 possible gametes. And I can see those using this Punnett square. So these will become my gametes for my dihybrid cross in the next section. Here's an example of how that dihybrid cross would be set up. I have my 4 gametes, and since both parents were the same on the previous slide, I can use the same gametes on both sides. The top and the left will come together to form the F1 offspring. In this case, capital Y, capital R. You're always going to keep your letters together and you're always going to put capitals before lower cases. So I can see here, for example, even though my top would be a lowercase, my left is a capital Y. So I put the capital Y before the lowercase y, and I put all the y's together, and I do the same with the r's. I would take a second to try to fill in these other boxes to make sure you understand what you're doing. Here's the completed dihybrid cross. Please check it against what you did to make sure that you got this correct. Now, when you actually go through this dihybrid cross, you could calculate genotypic ratio, but it's going to take you a bit of time and it's not really going to help you very much. What I want you to see here is the phenotypic ratio. So if you look on the right, you'll see that there are 4 possible phenotypes: yellow round, yellow wrinkled, green round, and green wrinkled. The spaces mean it doesn't matter what allele is there, because I have a dominant, it could be recessive, it's still going to be yellow, it's still going to be round, be it as may. Now, if you have set this up so your capital is first and your lowercase is last, you will see a very particular pattern. Your yellow rounds will be these, your yellow wrinkled, your green round, and your green wrinkled. So what we can see here is that there is a 9 to 3 to 3 to 1 phenotypic ratio. 9 that are dominant, 3 that are recessive for one trait, 3 that are recessive for the other trait, and 1 that is recessive for both. In a dihybrid cross, where both parents are heterozygous, I will always see a 9 to 3 to 3 to 1 relationship. Both of Mendel's laws reflect rules of probability. Beyond a dihybrid cross, it's not really practical to keep doing Punnett squares. So instead, what you can do is you can use statistics, you can use probability, you can use math to figure this out. Now, there are 2 rules here, the rule of addition and the rule of multiplication. We've been doing the rule of addition the whole time because, say, homozygous dominant and heterozygous purple, we're saying there's 3 purple offspring. You have to add that up, even though they're different. So, rule of addition, you have an event that can occur in more than one way. The rule of multiplication says, well, if I have independent events, what are they likely to do together? So if you do monohybrid crosses for each trait separately, you can get your math. So I know that if I have capital Y lowercase y crossed with a capital Y lowercase y, I'm going to have 3 out of 4 being yellow. If I do the same for round, I have 3/4 times 3/4, which is 9/16, which I just saw on my dihybrid cross.